The contractile filaments within the sarcomere are primarily composed of two proteins, actin and myosin, and are attached and held in between Z-discs. Actin filaments form thin filaments which contain helixes of tropomyosin and G-actin. G-actin has an active binding site that is revealed when there is a conformational change caused by the troponin. The tropomyosin also have a triple bundle of troponin which contain troponin C, I, and T. Myosin filaments form thick filaments. These filaments slide past each other during muscle contraction, facilitated by molecular interactions between the actin and the myosin. Starting with action potential of muscle contraction, we have a motor neuron at the site of the muscle. The neuron has a voltage-gated sodium channel that will allow sodium in and open voltage-gated calcium channels. As calcium rushes in, it activates a snare protein that will then cause the release of acetylcholine. The acetylcholine then binds to ligand acetylcholine channels that will allow sodium to pass into the cell, along with the voltage-gated sodium channels. The sodium will then create an action potential that will activate the dihydropyride receptors, causing them to open and allowing calcium to flow out of the sarcoplasmic reticulum. The calcium then attaches to the troponin, which in turn causes a conformational change that will reveal the G-actin active site. This will allow the myosin head to attach to the actin, starting the cross-bridge cycle. Once the head is attached, it releases heat that will then be used to lose the phosphorus molecule attached to it. And then following this, it will lose the ADP and move the head from a cocked to an uncocked position, causing a contraction of the muscle. ATP is then used to detach the head from the actin, and then it will go through hydrolysis, binding to a phosphorus molecule, which will return the head to a cocked position with ADP and phosphorus ready to repeat the cycle. Regarding sources of ATP, we have anaerobic sources, or ones not needing oxygen, or aerobic, needing oxygen. In the anaerobic category, we have the cytosolic ATP found in the cytoplasm of the cell. It will provide about 5 seconds of energy and is perfect for short bursts of movement. Creatine phosphate is another which only provides 10 seconds of energy and is good for short bursts of high-intensity activity. Glycolysis is another form that uses glucose to create ATP and will provide 60 seconds of energy and is good for high-intensity activity when oxygen availability is limited. Aerobic ATP comes from the mitochondria that utilizes oxygen in the production of ATP and can provide hours of energy, which is good for prolonged activities. Smooth muscle is categorized into two main types: single unit and multi-unit smooth muscle. Single unit smooth muscle are linked together via gap junctions that allow them to contract as a single unit. Contractions of this sort often spread as waves, starting at one end and spreading to the other end. Multi-unit smooth muscle consists of individual muscle fibers that are not electrically coupled. Each fiber functions independently and is innervated by its own motor neuron. Multi-unit smooth muscle is found in structures requiring fine control, such as the iris of the eye and the erector pili muscles in the skin. For the molecular mechanism of muscle contraction in smooth muscles, we start with calcium entry. Similar to skeletal muscle, the initial step in smooth muscle contraction is the entry of calcium ions into the cytoplasm from extracellular space, such as for the tonic cells, and then from the intracellular stores in the sarcoplasmic reticulum for the phasic cells. Calcium binds to the protein calmodulin to form a calcium calmodulin complex, which activates the MLCK in the cell, which phosphorylates the myosin head, leaving it with two phosphoruses. Phosphorylation of the myosin head activates the myosin ATPase activity, allowing the myosin head to bind to the actin filaments and initiate the cross-bridge cycle. For phasic cells, the cycle follows by losing a phosphorus, then ADP. Unlike skeletal muscles, though, a phosphorus is left over. This process then moves the head from a cocked to an uncocked position. ATP is then introduced, unbinding the head from the actin. From here, there are two paths it can go. It can go to relaxation or continue the cycle. If it relaxes, it will introduce MLCP, which will remove a phosphorus and then go through hydrolysis to return it to a cocked position with ADP and one phosphorus. If it follows the other path instead, it will skip MLCP and just perform hydrolysis to add the ADP and a second phosphorus to allow it to follow the cycle again without needing MLCK again. For tonic cells, it will follow by losing a phosphorus, then ADP, allowing the head to move to an uncocked position. Then MLCP will be introduced earlier, removing the phosphorus, making it harder for the ATP to detach the head, which is good for muscles such as sphincters that are held closed for a long time. As ATP is introduced, it will take much longer for it to latch, which is represented by the squiggly arrow labeled latch stage. Once it does, it will go through hydrolysis and leave the head prepped for the cycle again with ADP and a phosphorus. Structurally, smooth muscle lacks striations due to the arrangement of actin and myosin filaments in a more irregular pattern compared to the highly organized sarcomeres of the skeletal muscle. Smooth muscle cells are spindle-shaped with a single nucleus, whereas skeletal muscles are multinucleated and cylindrical. Functionally, smooth muscles exhibit slower and sustained contractions compared to the rapid, forceful contractions of skeletal muscles. Smooth muscle is also capable of stretching more than skeletal muscle, allowing for greater elasticity in organs like the bladder and blood vessels. Regulation of smooth muscle contraction is influenced by both neural and hormonal factors, as well as local factors such as stretch and chemical changes in tissue environment. In contrast, skeletal muscle contractions are mainly regulated by the somatic nervous system via motor neurons. Additionally, smooth muscle contraction can be initiated by pacemaker cells or by stretch, whereas skeletal muscle contractions are primarily voluntary and initiated by motor neuron stimulation. Mundle spindles are sensory receptors embedded within the skeletal muscle fibers, sensitive to change in muscle length and the rate of change of the muscle length. They consist of specialized muscle fibers called intrafusal fibers, surrounded by sensory nerve endings. When a muscle is stretched, the intrafusal fibers within the muscle spindle stretch as well, leading to depolarization of the sensory nerve endings. This sensory input is transmitted to the spinal cord, where it contributes to the regulation of the muscle tone and coordination of the movement through reciprocal inhibition. This is where a muscle, such as the bicep, are active, which inhibits the activity of the triceps. Golgi tendon organs are located at the junction between the muscle and its tendon. They detect changes in muscle tension or force rather than changes in muscle length. Golgi tendon organs consist of sensory nerve endings entwined with collagen fibers of the tendon. When the muscle tension increases, collagen fibers in the tendon are stretched, leading to activation of the Golgi tendon organs, sensory nerve endings. This information is transmitted to the spinal cord and contributes to the regulation of muscle force and prevention of muscle damage. Stretch reflex is a monosynaptic reflex arc that involves muscle spindle sensory receptors, the spinal cord, and muscle fibers. When a muscle is stretched, the muscle spindle sensory neurons transmit signals to the spinal cord, where they synapse directly with alpha motor neurons innervating the same muscle. This results in a rapid contraction of stretched muscle, known as the stretch reflex. The stretch reflex helps maintain muscle tone and stability by automatically adjusting muscle length in response to sudden changes. An example of this is when you're falling asleep, your head will fall to one side, stretching the muscle, which will send a signal to the spinal cord. It will then activate the muscle on that side, returning it to its original position. The withdrawal reflex is a polysynaptic reflex arc involved in removing a body part from a noxious stimulus. It involves sensory neurons detecting the noxious stimulus and transmitting signals to interneurons in the spinal cord, which then synapse with motor neurons. The motor neurons then activate the appropriate muscle to withdraw the body part from the stimulus. An example is the withdrawal of the hand from touching a hot object. Chemoreception involves the detection of chemical stimuli by sensory receptors. In smell, chemical molecules in the air bind to the olfactory receptors in the olfactory epithelium of the nasal cavity. These receptors are specialized G proteins that initiate an action potential in the olfactory nerve fibers, transmitting the signal to the olfactory bulb in the brain for processing. In taste, chemical molecules dissolve in saliva, bind to taste receptors located on taste buds on the tongue and other parts of the oral cavity. There are 5 primary taste qualities: sweet, sour, salty, bitter, and umami. Activation of taste receptors triggers the release of neurotransmitters, leading to the generation of action potential in sensory neurons that transmits taste signals to the brainstem and then to higher brain regions for perception. For sound waves changing into action potential, it involves the conversion of the sound wave into mechanical vibrations, fluid waves, and ultimately action potentials in the auditory neurons. Sound waves are collected by the outer ear and funneled into the ear canal. These waves cause the eardrum to vibrate. The vibration of the eardrum transmitted through the middle ear oscillates, which amplifies and transmits vibration to the oval window of the cochlea. The vibrations of the oval window cause fluid waves within the cochlea, specifically within the cochlear duct containing the organ of Corti. Hair cell activation, or hair cells within the organ of Corti are bent by the fluid waves, leading to the opening of ion channels and generation of receptor potentials. Receptor potentials trigger the release of neurotransmitters, stimulating sensory neurons to generate action potential. These action potentials are transmitted to the brainstem and then to higher brain regions, where they are processed and perceived as sound. The process of focusing light in the eye involves several structures and mechanisms. Light enters the eye through the cornea, which is the transparent outer covering of the eye. The cornea helps to refract or bend light rays towards the lens. The lens, located behind the iris, is a transparent, flexible structure that further refracts the light to focus it onto the retina. The ciliary body is a ring of smooth muscles located behind the iris. It controls the shape of the lens to adjust its focusing power. Suspensory ligaments connect the ciliary body to the lens. When the ciliary body contracts, it releases tension on the suspensory ligaments, allowing the lens to become more rounded for focusing on nearby objects. When the ciliary body relaxes, the suspensory ligament pulls on the lens, flattening it for focusing on distant objects. The retina is the innermost layer of the eye that contains photoreceptor cells. There are two main types of photoreceptors, rods and cones. Rods are highly sensitive to light and function well in low light conditions. They are responsible for peripheral night vision, but do not provide color vision. Rods contain the pigment rhodopsin, which undergoes a chemical change when exposed to light, leading to generation of electrical signals. Cones are less sensitive to light but are responsible for color vision and visual acuity. There are 3 types of cones, each sensitive to different wavelengths of light corresponding to red, green, and blue colors. Cones contain photopigments that undergo a similar chemical change when exposed to light, generating electrical signals that are transmitted to the brain. Receptors all have the same makeup inside. First, in the center, we have a G protein that contains a bent 11-cis retinol protein. When light hits this protein, it straightens out to become the 11-trans retinol that will then activate the G protein. It will then activate the PDE receptor that will then close off sodium channels and reduce the cGMP in the cell, which leads to a reduction in the release of glutamine to the bipolar cell. The surrounding has the same process, but instead releases glutamine to horizontal cells that have AMPA receptors that are stimulated by glutamine and activate the release of GABA, which inhibits the use of glutamine in the bipolar cell. On-center off-surround cells contain a mGluR6 receptor on the bipolar cell that when glutamine is activated, it will close off the calcium channels, reducing action potentials. As light hits the center of these, it will reduce the glutamine released, creating 60% of the action potentials. The surrounding is responsible for the other 40%, 20% on each side. When the light hits the surrounding, it will also reduce the glutamine released, which will slow down the release of GABA in the horizontal cells, preventing inhibition of glutamine to be absorbed, which would leave only the 60% action potentials total if light was on both the surround and center. With off-center on surround, the bipolar cell has AMPA receptors that are stimulated by glutamine. When light hits the center, it will again reduce glutamine release, which won't activate the AMPA, which will not lead to action potentials. When light hits the surround, it will again lower the release of glutamine, and the horizontal cells in turn will not release as much GABA, allowing for more glutamine in the center to be absorbed by the bipolar cell, leading to 40% action potentials, again 20% on each side. These action potentials will release glutamine from the bipolar cell to the ganglia, which will send a signal to the brain for perception of that image, of that light.